Good morning. It's October 17th, and this is your daily brief in science. Here's everything you need to know. A new discovery of a 230-million-year-old dinosaur fossil from Argentina is reshaping our understanding of dinosaur evolution. The fossil, named dyrk-kinases-regulate-cilia, was found in the Andes mountain range and includes a nearly complete skeleton, parts of the skull, the entire spine to the tail, and most limb bones. This herbivorous dinosaur lived at the end of the Triassic period, measuring about 2 meters long and weighing roughly 18 kilograms. Its anatomical features suggest early adaptations related to foraging and browsing, providing clues about the evolution of neck elongation and gigantism in sauropods. The discovery indicates that the development of long-necked sauropods began earlier than previously thought, bridging a gap between small-bodied Carnian sauropodomorphs and larger Norian stage genera. It also offers insights into the ecosystem's complexity during the late Triassic, where early dinosaurs coexisted with etosaurs, rhynchosaurs, and cynodonts. The fossil's geological context provides a detailed framework for studying evolutionary rates during a period of climatic fluctuations. This find challenges existing timelines, suggesting that giant sauropods like Argentinosaurus may have origins over 100 million years earlier. It expands the known geographic distribution of dinosaur faunas in Gondwana, indicating diverse ecosystems across different regions. Discovered in 2018 at an altitude of 3,000 meters in La Rioja province by a CONICET team, the site's environment underscores its scientific importance. The discovery was published in the journal Nature, highlighting the significance of South American fossil sites like the Isquigualasto Formation for understanding early dinosaur evolution. A recent study from the University of Texas at Arlington, published in Current Biology, uncovers a new role for mitochondria beyond energy production. The research shows that mitochondria also help protect cells during death by absorbing calcium, which is crucial for the process known as apoptosis. The study found that when certain caspases, the enzymes that promote cell death, are absent, cells can sometimes recover from early death signals. This indicates that apoptosis can be reversible or abortive at certain stages. Mitochondria transported by kinesin 3 help prevent localized calcium spikes that activate caspases, thereby blocking specific cell death pathways. The research team used live imaging and super-resolution microscopy on embryonic cells of C. elegans to observe mitochondrial behavior in real time. Led by doctoral candidate Rashna Sharman, the study highlighted how mitochondria function during both cell death and recovery. Understanding these mechanisms could impact brain development research and lead to targeted therapies for diseases like cancer and neurodegenerative disorders. The study was supported by the Cancer Prevention Research Institute of Texas and the NIH, and involved experts like Mark Pellegrino and Pia Gose. Overall, the findings expand our understanding of mitochondria as key regulators of cell survival and death, revealing their complex roles in cellular biology. Researchers at Tokyo University of Science have developed a fiber-coupled single-photon source that generates photons directly inside an optical fiber. This innovation aims to reduce transmission loss and increase efficiency for quantum communication systems. The system works by selectively exciting a single neodymium ion within a tapered silica fiber doped with neodymium 3+ ions. This process allows for the direct generation and guidance of single photons at room temperature. Operating at telecom wavelengths, the device is cost-effective and compatible with existing fiber networks. It does not require cryogenic cooling, making it practical for real-world applications. The research was published in Optics Xpress on September 22, 2025, and marks progress towards scalable quantum technologies. The technology improves single-photon emission efficiency, which is essential for quantum key distribution and secure communication. It minimizes losses and maintains optical quality after tapering. The use of commercially available fibers and doping techniques enhances the system's scalability and affordability. Future developments may focus on optimizing photon wavelengths for spectroscopy and imaging. This approach could also support quantum computing by enabling control of multiple ions within a single fiber. Experimental validation through autocorrelation analysis confirmed the system's ability to emit one photon at a time with high efficiency. Overall, this room temperature, cost-effective quantum light source advances practical quantum networks and scalable quantum information processing. Cancer cells can hijack genetic programs active during early development to support their growth. New research published in Nucleic Acids Research shows that these cells reactivate embryonic splicing factors, proteins that modify RNA to promote tumor progression. This reactivation allows tumors to acquire features similar to early embryonic stages, making them more aggressive and adaptable. The study found that cancer cells selectively re-express splicing factors from embryogenesis, rewiring RNA splicing to produce variants that favor growth. Targeting these specific splicing factors with drugs could potentially interfere with this process, offering a new approach to slow or stop tumor growth. Additionally, changes in splicing factor activity during early stages might serve as biomarkers for early cancer detection. Researchers utilized artificial intelligence to analyze gene activity patterns, enabling them to identify reprogramming of splicing factors more efficiently than traditional methods. This combination of molecular biology and AI revealed vulnerabilities in cancer's gene regulation networks. Oncogenes like MYC play a role by disrupting the balance of splicing factors, amplifying growth-promoting variants while suppressing inhibitors. The activation of MYC triggers a cascade that shifts healthy cells into a malignant state, highlighting how cancer rewires its gene-editing machinery to sustain proliferation. Primary cilia are tiny, antenna-like structures on cells that play a crucial role in sensing the environment and transmitting signals essential for growth and development. Recent research led by Professor Dr. Elif Nurfirat Karalar at Koç University has identified the dyrk family kinases as key regulators of these structures. The study shows that when dyrk kinases malfunction, primary cilia can become abnormally long, deformed, or unstable, impairing the cell's ability to detect signals accurately. The activity of dyrk enzymes is necessary to maintain normal ciliary structure. Without it, defects and instability occur. These cilia are involved in sensing signals such as hedgehog, WNT, and PDGF, which are vital for cellular homeostasis and development. Disruptions in ciliary function have been linked to various health issues, including developmental disorders, kidney diseases, and vision loss. The research also provides insights into potential molecular targets for treating ciliopathies and related diseases. Additionally, since ciliary signaling influences tumor growth and metastasis, modulating dyrk activity could have implications for cancer therapies. Professor Karalar's background includes a PhD from UC Berkeley and postdoctoral work at Stanford, with her lab focusing on cytoskeleton and organelle dynamics. The study employed genetic manipulation, live cell imaging, and biochemical assays to understand how dyrk kinases affect ciliary structure and function. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. We'll be back Monday with everything you need to know.